Okay, so, gentlemen, today we are going to start the discussion of chapter 15 and the chromosomal basis of inheritance. Um, Remember that Mendel had no idea how his hereditary factors were being passed on from one generation to the next. He had no idea about what the structure of DNA or even about chromosomes for that matter. And so, how these hereditary factors were passed on from parent generation to first filial or second filial generations was a mystery, although he knew that they were being passed on. So, it turns out that today we actually can demonstrate that the factors that Mendel talked about are genes, all right, and they are located on chromosomes. As a matter of fact, we can even isolate particular genes on a chromosome inside of a cell using very specific dyes that attach to specific genes. Right? This is an awesome picture of. I don't know why that happens. This is an awesome picture of a pair of homologous chromosomes with a very specific gene highlighted here and here. All right, and this is the kind of thing that we have the ability to do today. This is an early drawing of mitosis from the late 1800s. And this is when people started first realizing that maybe chromosomes were part of the whole idea of inheritance. In the late 1800s, biologists, especially cell biologists, worked out the processes of mitosis and meiosis using compound light microscopes, just like we can use today. Biologists then began to see that perhaps there were parallels between Mendel's ideas, his hereditary factors, and the chromosomes that they were seeing under the microscope. And in the early 1900s, we began to develop what's today known as the chromosomal theory of inheritance. In this diagram, we illustrate both of Mendel's major laws here. On the left, we have the law of segregation. And if you look at the left side of the diagram, starting here with the original cell, notice first the cells duplicate, then they divide in anaphase 1 and they produce these two haploid cells. The haploid cells then undergo meiosis 2 and produce these 4 gametes that you see down here. And so what it says is that the 2 alleles that each organism carries are separated, all right? Notice I've got a big Y over here and a big R and a big Y and a big R and a little Y and a little R and a little Y. So the Y's and the R's, the big Y and the big R, the big Y and the little Y are separated. The big R and the little r are separated. On the other side, we show, gosh darn it, we show the law of independent assortment. Same original cell, but this time the maternal and paternal chromosomes line up on opposite sides. And so after going through meiosis 1 and 2, we end up with a set of gametes that looks like this. Now notice, here we've got still the Y's, the big Y and the little Y are separated, the big R and the little r are separated, but it's now big Y and little r together and little y and big R together. All right. And so this is the process that leads us to the 9 to 3 to 3 to 1 ratio that Mendel was finding in his studies. Okay. So, looking at this, we know what the law of segregation and the law of independent assortment are, but because of our knowledge now of genes and chromosomes, we can actually demonstrate how these things actually work. So who was it that actually came up with the idea for all of this? The answer is a guy by the name of Morgan. In the early 1900s, Joe Morgan did a great deal of work that eventually led to the conclusion that chromosomes were the physical basis of inheritance. All right. So how did he do this? Morgan showed that Mendelian inheritance has its physical basis in the behavior of chromosomes. All right. the first solid evidence that he associated a specific gene with a specific chromosome came in the early 1900s from the work of Joe Morgan. His early experiments provided convincing evidence that chromosomes are the location of Mendel's heritable factors. So, how did he do this? Well, Morgan used the common fruit fly, Drosophila melanogaster, as his study species, and there's some good reasons for this. First, okay.
It's 8 o'clock.
Thank you, Jesus. I'm not really sure what this is about, but okay. Now I'm starting to get really annoyed. Are we done? Oh, good. So Morgan's choice of, I don't know, I have no idea what will happen. My ghost in the machine. Anyway, fruit flies have several advantages for this kind of thing. First, they produce lots and lots of offspring. All right, one pair of fruit flies can give rise to hundreds in even just a couple of weeks, and every generation, each generation can be produced within 2 weeks of each other. And finally, there are only 4 pairs of chromosomes in a fruit fly, and therefore this makes it fairly easy to follow inheritance patterns. So Morgan noted that there were wild-type fruit flies were the most common fruit fly, and the fly on the left is the one that is wild-type. But there are traits out there that are alternative to the wild type, and we call these the mutant phenotypes. Now, the first mutant that Morgan identified was a white-eyed individual. Look at this guy, all right? Funky-looking white little eyes instead of the wild type red eyes, all right? And so this is the mutant or alternate trait for eye color, all right? So 2 alleles, red eye, white eye, all right? But they're both the eye color gene. Okay, so in one experiment, Morgan took male flies with white eyes, the mutants, and he mated them with female flies that had red eyes, the wild type eyes, okay? And you can see that original experiment here. So this is the P generation. Here's the red-eyed females. Here's the white-eyed males. He found that all of the offspring had red eyes, okay? Every single one. But then he noted that in the F2, he found that there were 3 red-eyed organisms to 1 white-eyed. So it's a 3-to-1 ratio. Great, all fine and good. But there's one little catch here. He noted that every single one of the white-eyed flies was male. Now, what that led him to believe was that the gene for eye color must be located on the X chromosome. And so therefore, if you had the recessive white-eyed allele and you were a male, you were white-eyed automatically. If you had a recessive white-eyed allele and you were a male, but you also had a red-eyed dominant allele, then you were red-eyed. And of course, the females, because they had 2 X genes, I'm sorry, 2 X chromosomes, and therefore 2 alleles, they had the red eyes as well. All right. Now, Morgan's work supported the chromosomal theory of inheritance. Now, the conclusion for today is shown on the right-hand side. All right. The the red-eyed females had 2 copies of the wild-type gene. The white-eyed males had 1 copy of the recessive gene. They produced their gametes, and these are the possible combinations of offspring in the F1 generation. All the females were going to be red-eyed because they had the red-eyed allele, and all the males were going to be red-eyed because they had the red allele. However, when these males and females then produced offspring, the The results were that all of the females end up with red eyes because each one of them carries at least one copy of the red-eyed allele. The males had a 50-50 shot at becoming red-eyed or white-eyed because half of the males would have a red-eyed allele and the other half would have the white-eyed allele. Okay, so Morgan was able to successfully demonstrate the chromosomal theory of inheritance. All right.